Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In this class we will be talking about data QC and data trimming. Just to briefly recap, we have discussed data formats and followed by data quality control or data QC. So, in the last few classes we have talked about different aspects of data QC. We have talked about one tool that allows us to do this quality control. So, we will continue that discussion today. We will complete all the quality control measurements that we have, followed by we will talk about something called data trimming. Okay. So, this is a part of the pre-processing step, and this is a very important step before we go into the actual analysis. So, these will be the keywords we will be seeing today: sequence based control, quality trimming and contaminant trimming. So, this is the tool that we have discussed in the last class. So, the tool is FastQC. So, this is a free tool that we use to evaluate the next generation sequencing data. So, this tool allows us to check quality only for FastQ data sets, right. So, for example, Illumina data sets, right, or some other data sets that can be converted to FastQ format. Okay. So, some of the controls we have talked about already, and all of them are based on quality score, right. So, this is something that we have discussed in the last class. We talked about, for example, par read quality or par base quality, right, and tile quality, all those things we have discussed, and they rely on this quality score, right. So, this quality score comes up the machine, it measures the signal to noise ratio, it gives a measure of confidence in the base call. So, there is another aspect of quality control that we will be talking about today, and this is the sequence based quality control. Okay. So, compared to Q score based control, here the FastQC tool actually looks into the sequence of the data, right. So, whether the data sequence actually makes sense. So, maybe the quality score is very good, but nevertheless you can have some sort of contaminant, right, or some sort of DNA molecules that are not supposed to be there, right. So, they will not show up in the Q score based control, they will appear only when you do the sequence based QC, right. So, let us get started on that part. Let us talk about the sequence based QC in much more detail. So, one of the first parameters that we check is called sequence length distribution, right. So, this is something that is applicable to all platforms that you work with. Here this is an example with Illumina data, right. So, in the last class we are seeing this example with Illumina data. So, in this example we are again seeing this Illumina data. Along the x axis you have the sequence length, and along the y axis you have the number of reads. Okay. So, what does it show? It shows the distribution or the number of reads that have the certain sequence length, okay, or read length. So, here because this is Illumina data, all the reads are of same length, right. So, which is the 125 base pair here, right. So, if you remember the principle, Illumina will give you reads of same length. So, that is what we see here, we have 125 base pairs. For other platforms, right, this will vary, right, you will not see this kind of distribution where all reads are of same length. You can see different distributions of read lengths, right. So, what is the purpose of this? It is like that is to check that we have got good read data, right. We have at least a certain length of reads, right. The second measure that we will talk about is something called par base n content. Okay. So, what does this measure? So, n means this is an ambiguous base. The sequencer was not able to resolve which base it is, right. So, it does not know whether it is A or G or C or T. It knows that there is a base, but it is not able to call the base properly because the signal to noise ratio is not very good. Okay. So, this is something that we want to minimize in our data, right. We do not want many n values in the sequence data, right. So, this is something we will see, right. So, what you have in x axis you have the position in read, right, this is in base pairs, and along y axis you have the percentage of n, right. And in this data what you see is there is a line hugging close to 0, right. So, that is a very good sign, which means we have very few n bases in the data, right, and this is a very good sign that the sequencing was in general good. you have got very good data, because there is not much end in your data set. Okay. Also remember, this will also appear in the quality score based QC, right. So, you have, because any position of n will have low quality score, because the sequencer is not able to resolve this base properly, and this is something that will appear in both, right. If you are looking at the sequence based quality control or Q score based quality control, this will appear in both. The next measure is the GC content, right. So, you have studied what is GC content, right. So, this is the percentage GC base in the genome of an organism, right. So, some organisms may have high GC bias, some organisms may have AT bias, right. So, this is something that is there. So, what this tool does, it gives you a distribution, observed distribution of GC content in the reads. Okay. So, this is something what you see here in red. So, the red distribution is the observed distribution, right. So, along x axis you have mean GC content, right. So, for this data it is around 40%, right. You see this point here, right. So, it is around 40%. Again, this can vary from one organism to another, and along y axis you have the number of reads. Okay. Then you have the blue line, okay, which is the theoretical distribution. So, this is a distribution that is actually generated by the tool based on the data. Okay. Remember, the tool does not know which organism is being sequenced. So, it does not have any idea about the expected GC distribution, right. It simply generates this theoretical distribution from what it sees in the read data. Now, this looks fine, it means it shows like the theoretical distribution kind of matches with the observed data, and most importantly this is a normal distribution, right. So, any departure from normal distribution would suggest there is something wrong, or something else has happened with the data set, right. Now, this distribution can shift towards left or right depending on the organism that you are working with. So, this is something that you can also check yourself, right. Once you know, okay, my organism should have this percentage of GC content, you can check back and see whether you see that percentage here in this plot. Okay. This kind of like a validation that you are actually getting the right organism data, right. Sometimes as we have discussed, during library preparation you can have contamination from other organisms, right. For example, you are working with primate genome, primate samples, and maybe you got some human contamination as well, or maybe you are working with yeast sample and you got human genome, and if these two samples have different kind of biases, right, GC biases, maybe they will show up here. Then the next measure is power based sequence content. Okay. So, this is something that is again looking at position wise GC and AT content. Okay. So, the earlier measure was overall GC content, right, in the read. This is power based GC content, right. So, power based sequence content, again looking at each position you have this percentage of AT GC, right, in 4 different colors. You can see the legend here, right. In 4 different colors you have this percentage AT GC, and here this kind of matches with what you have seen earlier, right. What we see for each position, the GC content is about around 40%, right. So, G is about 20%, C is also about 20%, and so GC content is about 40% and AT content is about 60%, right. So, in the earlier distribution we also saw that this GC content was about 40%. Okay. So, the initial part maybe you might want to see, right, this initial part you see like a lot of fluctuations, right, and as we have discussed, right, when we are discussing the quality control, data format, etc., we have discussed, right, in the beginning of the sequencing in Illumina platform you have this low quality score, right, because this is where the sequencer is kind of identifying the clusters, setting the thresholds for signals, etc. Okay. So, in this part this kind of fluctuates a lot, but this is that we do not have to worry about too much. Rest of the data looks fine to us. So, there could be different types of duplicate sequences, and we will have a look now what are the different types that you can get especially in Illumina platform. So, one is optical duplicates, where a single cluster is mistaken as 2 clusters by the detector. So, this is something that is kind of a problem with the detector, which instead of identifying a single cluster, it identifies them as 2 clusters. So, we get the same sequence data for these reads. Then you can have clustering duplicates, where the same molecule goes and occupies two adjacent clusters during the cluster generation process, right. So, again we do not have control over this cluster generation process exactly, right, which molecule will go where. Now, what this might happen, right, you can have same molecule going to two adjacent clusters and they will again give rise to the same read sequence. Then you have the PCR duplicates. So, this is present in all types of platforms where you need to do amplification, right. This actually arises because some molecules are preferentially amplified and then these molecules then go and occupy different cluster locations. And finally, sister duplicates. So, these are actually class of duplicates where complementary strands of the same molecule are found in independent clusters. Now, you might ask like whether these are actually duplicates. So, some programs or tools will mark these as duplicates, whereas some would not mark these as duplicates because they are complementary strands, which means we are sequencing from two different directions. Okay, so what is the problem with these duplicates, right? So, duplicates means they are coming from the same molecule, right? They are not independent molecules that have been sequenced. They are same molecule sequenced multiple times. Now, what would be the problem? So, one of the first problem is that we have problem with detection in single or multinucleotide variants. Okay, so we will discuss this in a lot more detail when we talk about this single nucleotide variant detection, etc. Right. So, the problem starts is like if you have a read that has a mutation compared to the reference sequence, right, and if that read is duplicated, right, or present in multiple copies, right, you might say okay, this mutation appears to be very dominant across my DNA library, right. In reality, this could be something else, right. This mutation may be present at very low frequency in the library, but may appear as a very highly frequent mutant in the library, right. So, this is something we will discuss. So, this can happen, right, and also this can cause problems in detection of the actual SNVs or MNVs. And as I mentioned, you also have problems with calculating frequencies of these variants, right, actual frequencies, because duplicate might alter, may kind of inflate the frequency in some cases. It might also cause problem in copy number variation analysis. Again, copy number variation, we are looking at whether there is any change in copy number of a gene or a segment of genome, right. But then if you have too many duplicates for that region, they might appear as amplified or with increased copy number, right. So, this is something we do not want, we want to avoid this kind of situation. Okay, so, we will discuss like how we actually go about it and what could be the actual errors when we have this duplicate data. Right. So, one of the things we do, right, so coming back to this duplicate sequence and duplicated sequences, right, this is something that we have to worry about, right, and we have to pre-process the data if we actually can do that, right. So, before we actually go into the downstream analysis. So, what is also important to note is that this is not really that important for RNA-seq data analysis when you are looking at transcriptome data analysis, because in transcriptome analysis it is expected if you are sequencing at a very high depth, you are doing deep sequencing, it is expected that you will get duplicates, right, because in transcriptomics you are looking at only limited part of the genome, only a small part of the genome, right, the protein coding or mRNA coding regions, right. So, these regions are kind of limited and you can get multiple copies of the same fragment. Okay. So, in RNA-seq we are usually not doing this duplicate analysis or duplicate removal. Okay. Another problem that might occur, and this is something that this tool will look at, is something called overrepresented sequences. Okay. So, NGS libraries, you can imagine, right, we are working with big genomes where you have fragmented this DNA into small fragments and they are likely to be very diverse, right, and you should not see a single read sequence at very high frequency and occurring multiple times in the library, right. And this kind of problems might occur, right, and this program will identify whether there are overrepresented sequences, right, and if they are present, this might mean that there is something relevant biologically, right. There is some important biological phenomenon that has happened that has enriched certain parts of the genome or certain parts or certain genes, etc. Second, it could mean there is a contamination, right. There could be a large scale contamination from other fragment into your sample. So, you may have to be careful when you are processing the sample. And number C, maybe the library preparation was not good enough, probably many of the genomic fragments were lost during library preparation. So, the library is not as diverse as expected. Okay, so, these are the things that you have to worry about. The third point of course, is something that will require again library preparation and sequencing. The first problem is something interesting and relevant. The second problem we can deal with through computational tools. Okay, so, we will go forward and see how we can do that. The final point that this tool will look at is something called adapter content. Okay. So, you remember these adapters that are added at the end of DNA fragments while we are doing sequencing, right. So, these adapters could be different, right, and usually are different. And when what you see here, this is a plot that is generated by this tool, is along x axis you have position in read, again certain length, and y axis you have percent of all reads. And this program will check whether it finds some adapter sequence match with certain adapter sequences in this read data. Okay. And here in this data set you can see there is some adapter content or adapter contamination. Okay. This black line, it actually goes up, right, as we go towards the end of the read, the percentage of reads showing this adapter sequence, it actually goes up quite high, right. It is about 40% of the reads actually have adapter contamination. And this is something that is a problem. It also shows you the type of adapter that is actually contaminating the data, right. So, in this case this is the black line means this is a next era adapter that was used for this library preparation, and the other adapter sequence they are not present, right. You can see this one in purple, the solid small RNA adapter, right. So, this is not present, but the tool could find this next era adapter in the data. Okay. So, this is again a very important observation, and it means we need to actually pre-process data. Now, the question that you have is how does this adapter contamination happen, right? They are not supposed to be there in the read data, right? We should get the sequence of the DNA fragment or the genome fragment, right. So, one of the things that I will mention is that this adapter contamination depends on the length of DNA fragment that are present in the library. Okay. So, let us take this example and then we will probably understand how we get this adapter contamination. So, let us imagine we are doing Illumina 200 base pair run, okay, and we have prepared this genomic fragment and some fragments with these adapters, right. Some fragments are of length, let us say 500 base pair, right. So, here is the DNA fragment 500 base pair length, and you have adapter 1 on one side, adapter 2 on one side, right, on the other side, and you are sequencing let us say from one side. Okay. Now, once you sequence, right, you will sequence 200 base pairs from this side. So, this is fine, right, you will get this sequence within this DNA fragment. Now, imagine the scenario when this DNA fragment is actually small. Okay. So, remember when you are preparing this library, when you are fragmenting the DNA, we have very little control over the actual size, right, unless we are doing enzymatic digestion. We cannot really accurately control the fragment that will be generated, right. So, this is something that kind of has been optimized with different trial and error. Okay. So, if your fragment length is small, if it is 100 base pair only, we are sequencing 200 base pair, let us say, right. So, what will happen? You start sequencing from this side, it will sequence this 100 and then it will go into this adapter 2, right, on the right side. So, this adapter sequence will come as part of your read sequence, okay. Because the fragment length is small, you end up sequencing the adapters, right. So, this can happen both in single end sequencing or paired end sequencing, right. So, if you are sequencing from the other end also, right, you will end up getting sequence of this adapter along with this yellow genomic fragment, right. So, this will give you this adapter contamination. Right. So, what is the impact of adapter contamination? Why are we worried about this? Right. So, it actually can impact the NGS data analysis, and as you will see the mapping quality suffers, right, because the adapter is a unique sequence and there is no match with the genomic DNA, right. So, the program that we will use for aligning these reads back to the reference genome, right, the mapping problem, this might not, the program might not be able to find this adapter sequence in the genome data, right. So, if it does not find, then it might get confused, right, what is happening, that there is no alignment, there is no mapping, and it might actually end up discarding the read from analysis. Okay. So, there might be some issues with the mapping. Okay. So, this is something that is a very important point, right, because mapping actually is the first step for a lot of downstream analysis, right. So, this is something you want to do very well. So, these are some of the quality controls that we have talked about in FastQC, but there are of course other quality control tools. For example, you have FastP, you have HT stream, etc., and again they have their own ways of doing things, they report different measures, but if you are interested, you can of course go and check these sites where you can actually find all the details about these tools. Okay. The next step, once you have done the quality control, right, so you have to do the quality evaluation, right. So, you have to once these statistics is generated, you kind of look at all these statistics and make decisions, right, how you should proceed with the data analysis. Okay. So, the tool FastQC will not do anything for you, right, it just shows okay, there might be problems here, there might be problems there, or this might be okay, etc. Now, you have to make this decision yourself, right, what you are going to do. Okay. And the first step in this is the data pre-processing. Okay. So, pre-processing actually consists of two steps. So, the first one is duplicate removal. So, as we have seen, if there might be duplicate sequences, and if the duplicate level is very high, you want to get rid of these duplicates probably first. Okay. So, this is something that we will do if it is required. If for example, if we are looking into single nucleotide variants or doing CNP analysis, we want to get rid of duplicates. The second step is something called data trimming. Okay. So, we will discuss what is data trimming in a moment. So, for duplicate removal tools, we have two different tools I will mention, but you can use other tools. There are multiple tools available, but we will focus on maybe one or two tools in this course. So, one tool is Picard, right. So, again I am giving the link so that you can go and check, you can read about the tools, how it works, etc. All these details are available. And there is a function in this tool. So, if you are familiar with Linux commands, fine, you will find this function mark duplicates function in this Picard tool. If not, do not worry, we will discuss how you run these Linux command lines and how you run all these tools. So, you have these options mark duplicates, remove duplicates. So, you can remove these duplicate sequences, or you can use the command called mark duplicates minus remove sequencing duplicates. Okay. So, they have similar functions. The other tool that you can use is called SAM tools. So, here again you have the link, and again it has some function called markdup function. So, if you want to remove duplicates, so markdup function it will just mark the duplicates, right. If you want to remove them, you have to use this command called markdup minus r. Okay. Again command line tools, how to write these shell commands, etc. we will discuss in the hands on. Now, coming to the next step, which is the data trimming, okay, and this is we would have to do, right. So, remember duplicate removal we may or may not do depending on the level of duplication, the kind of analysis that you are doing. If you are analyzing RNA-seq data, we may not worry about duplicate removal, etc. But if we have some sort of adapter contamination, etc., we will have to do trimming. Okay. This is a step that is required. Now, there are two types of trimming that you can do. One is called quality trimming and the second is adapter trimming. So, what is quality trimming? Right. So, this is removal of bad quality data without discarding the full read. So, we have discussed this kind of situation, right. So, imagine this situation where we are looking at this quality score, par-based quality score. X axis you have the position in read, y axis you have the quality score, and what you see towards the end, right, maybe beyond this point or so, the quality score drops significantly, right. So, you may not want to go ahead with the read this read data, right. So, what are the options? One is of course, let us discard this data for which you have this kind of low quality score, and this might end up kind of removing a lot of data, all right, that you have generated. The other option is maybe we can do something called quality trimming, right. Maybe we do not use this part of the read, right. Let us not use this right part, right. Let us trim this part of the read, only use the left part where the quality score is good, right. So, this is a good solution because you are not completely throwing out the read, you are actually working with good data that is available, right. So, this is the quality trimming. The other option is the contaminating sequences such as adapter sequence, right. So, we have seen this adapter contamination scenario, right, where we have this adapter on the right hand side towards the end of the sequence, right. And again we want to remove this adapter, right. So, again what this tool will do, the trimming tools will do, they will identify, okay, there is this adapter sequence and I should remove these adapters, right. So, this is where this trimming is important. So, the example that we have taken, we need to do adapter trimming, we need to remove these adapter sequences. Okay. And the importance of trimming is that we get an overall improvement in data quality, right. So, if you check Q score etc., sequence based QCs, you will see this improvement and without much loss of good quality data that you have. Okay. As usual, there are many tools available. Again, we will use certain tools in our hands on class. So, you can go and check. Again, this one tool is called Trimomatic, right, and it can remove adapters, it can do this leading, trailing, low quality base removal. It can also remove certain bases in between in the read, right, inside the read if they are below certain quality score, and it can also remove reads completely that are shorter than 36 bases. So, there is another tool which is called the BBDuck, right. So, again it can do this quality trimming, adapter trimming, and these tools use different algorithms, different ways of trimming, right, and we will use some of these tools in our hands on. And finally, I just want to give you some certain links. If you are interested, just go and check. Again, people use all sorts of tools, right. You can pick anything and see which one works the best for you, right. They are slightly different from each other, they have same function, but slightly different from each other, how they implement these functions, and you have to apply to your data and see which one actually works the best, right. So, these are again tools, right. So, for example, you have SQL which does trimming based on quality score, or Skype which does trimming based only for adapter sequences. So, these are the references that I have mentioned in this class. So, to conclude, so we have looked into two types of quality control, right. So, we have looked into quality score QC, and we have also now looked at sequence based QC, and taken together, right, they give us an overall idea about the quality of the data, right. So, both of these QCs are critical for evaluating this overall quality of the data that you have generated, right, and this is a very important first step, right. So, you want to look at the data and you want to do all sorts of pre-processing that are necessary for your data, right. We have also seen, right, duplicate reads can have detrimental effects on some of the downstream analysis, right, and again this is something you need to decide whether you want to do duplicate removal and whether it is actually going to affect your results, right, depending on the level of duplication etc. Then we have seen that the adapter contamination happens when the fragment size is small, right. I have given you a schematic, right, how this works, how this happens, right, and this is actually a very common problem with many DNA sequencing projects. You will see this adapter contamination and hence the downstream pre-processing, right. So, the pre-processing where we remove duplicates and we also remove adapter contamination, these are required for correct inferences. So, these steps are really critical for all downstream analysis, right, whether you are going for genome assembly, whether you are going from for identification of mutants, whether you are going from transcriptomics etc. So, for any of this downstream analysis you want to do the first steps, which is the QC, then if required you want to do the pre-processing, and two steps of pre-processing that we talked about, one is duplicate removal and the adapter contamination. So, once you have done this, right, you are now good to go with the downstream analysis, and you can be sure, right, that whatever inferences that come out of this data analysis will be robust and accurate. Okay, so with that I will stop. Thank you.